I didn't get on with him because he was a communist, fellow traveller in those days, and I was very much against that. And I do remember when the Soviet invasion of Hungary in 1956, there was a meeting at the University College in Birkbeck by the Fabian Society, and Bernal spoke at that meeting. He had a few people at Birkbeck who were around him. They were sort of fellow travellers, were inspired by the great man. And he said, ever since June, this is 56, this is now October 56, ever since June, we've known that things have been going on in Soviet Russia. And I thought to myself, that June was the Khrushchev speech, 56. I thought, God, I've known this since 1938. I was 11 years old.
You mean the purges and everything?
Yes, purges. And so, but later I got to understand Bernal, and he knew a great deal about the past, but he believed that this would all pass, and just like he didn't use those words, but he looked at things with a long eye of history. And just as the French Revolution was a great upheaval, but it brought a new, well, it settled down, so to speak. There were a few other revolutions, 1838, 1848, and so on. But in the long eye of history, so that's how he viewed things. He believed in progress, you see. That's what he believed in. He believed in, and the book on science and the history of science, which was written, blowing rationalist for. But I was never friendly with him. He never inspired me, so to speak. But I did observe him, so to speak. But it was Roslin Franklin who made the difference.
Did she ever talk about? I mean, she was done down, as you mentioned, in the double helix.
Never complained. Never.
Never.
She just said, I could have kicked myself for not noticing that my photograph showed there was a twofold axis of symmetry. She didn't know enough crystallography. The whole story is really very strange, because when she was at King's, she had worked out the symmetry of the A form of crystal symmetry, and the crystal symmetry of the A form of DNA. She had sorted out the A and B forms, which was laid the basis for that work. Before that, people had been taking mixed DNA comes in two forms: A form, the B form. The B form is the one that we always see in flat discs, but the A form is a screwed up form of the B, which you get if you reduce the water, the vapor pressure, reduce the water content. And she had sorted this all out. She was a physical chemist. So so she had sorted out the two forms, the A and the B form. And the B form she knew was helical, and I said so. But the A form she thought was some opened out structure. The helix might have come apart. She didn't understand the picture. Now, what she should have realized was that the A form was also double helical, but it wasn't obviously so. And she simply missed it. She said, I could have kicked myself for missing the symmetry. That's what the word she said to me. And Watson recognized the relationship between the two forms when he saw the pictures, and they got hold of the report. You know, there's the famous report that was sent to all MRC units, which Crick and Watson seized upon, and they used her data. No question about that. So and to this day I don't know that she knew how much they knew of her work. But she should have shared the Nobel Prize with them. But then there was Wilkins around. It was not Wilkins. Well, Wilkins, you see in the story about Wilkins, Wilkins was rather kind of shy person, and I don't understand what went on. His line was, we must take our Rosie to dinner. give her a good talking to. It's very childish. I spoke to Roslin as an equal, and but you see, Wilkins was, but they never would have got on. They're very simple. He's clever. He's dead now. He's clever, and he had chosen DNA as a problem, which was, and but he wasn't. He had no kind of go in him, punch to go ahead. He thought he would, and Roslin was brought in not by him but by Randall, professor at King's, to put some muscle into the DNA effort. Because Wilkins was puttering around with ultraviolet spectroscopy and things like that, and imaging, and Roslin was a physical chemist. She'd never done crystallography before. So the irony is that Wilkins, Stokes, who was the theoretician at King's, and Roslin all had attended Bernal's courses in Cambridge in the 30s in crystallography. They'd all learnt about space groups, or they should have learnt about space groups, and none of them tweaked to it. Only Crick tweaked to it when he saw that this had the crystal symmetry of the A forms of the crystalline required that there must be twofold axes of symmetry within the molecule, and that's how he realized there must be a twofold. That's the double helix. And this came out only later when they wrote their paper in 1954. I'll give you a copy of my article on it.
One question about that particular incident. When I've talked to a number of scientists, they mention the name of a King's mathematician, John Griffiths, as important in all this work.
Yes, well, John was quite clever man. I know Francis writes about him. He went to a wine tasting.
That's right.
I used to know this all off by heart the night before. John Griffiths thought he paid attention to the fact that Chargaff's rules, or rather law, that the number of A's is equal to the number of T-adenine thymine. But they thought of them as stacking A's and T's, and A required a T in the chain. So he didn't get there. John Griffith had a distinguished father, Griffiths, who discovered DNA, but he didn't get. He was a clever man. Francis admired him, but they didn't get it, not until they knew about Roslin's work. Oh no, that had no influence upon the structure. Well, I can go on to this. Shall I give you an article on it? Which is 50 years to the day of the papers in Nature.
Yes.
The 3 papers in Nature. This is it. And it's an analysis of her. It's an analysis of the pictures, and explains. And I used her notebooks, which after she died, I found her notebooks, and I saw how close she had come. But she didn't have anybody, had nobody to talk to. Wilkins was here, broken.
If you'd been there, Aaron, just a bit earlier.
Oh yes, I would have seen it.
I'll get you to sign this later, if I may.
This shouldn't be capital D's, they put capitals in. And there's also 1 or 2. This is the aftermath. This is Kornberg. I was asked to.
But Hans Kornberg?
No, Arthur Kornberg, who just died, the father of Roger, who worked with me on, I know the word, I always used to know the dyad. A dyad is a twofold axis of symmetry. Twofold axis of symmetry means that you've got, these are 2 identical pens. So if you turn it 380 degrees, it looks exactly the same. Did you get that?
Yep, I got that.
That's how Perutz used to explain twofold symmetry. And there's the semi-conservative. There you are. And you want me to Just
signage, please. That would be lovely. Thank you.
Do you want me to say to you?
That would be very nice. Alan McFarlane. A-L-A-N.
No, I know. And McFarlane, are you with a small f?
Yes, I am.
And it's the one continuous word.
Yep, that's the one.
With compliments.
That would be
more
very
than
Cambridge.
best wishes.
Yeah, that's lovely. Lovely. Thank you very much indeed.
You know, it's sometimes said that Roslin didn't get on with Wilkins, and various reasons are given. I think they, even if he'd been a man, she'd been a man, it would have been the same story. Morris is shrewd, clever, but very, very slow. And any telephone conversation I ever had with Morris, and I had many of those, it would take half an hour to come to the point. Roslin was quick, decisive, and sometimes could be brusque, but impatient. And so they never would have got on. It's nothing to do with being a man and a woman. And so I think just wanted to get that clear.
Excellent. So after, how long did you work at
Birkbeck? I worked at Birkbeck I worked with her for 4 years, 1954 to 1958. And during those years we worked out the overall structure of tobacco-mosaic virus. And also I developed methods, analytical methods for turning the X-ray data on the films into making a map. Because of course the mathematics is rather different from that you use in three-dimensional protein crystals. So I wrote some papers, one of them with Crick, on how you do this. So, and then later, after she died, her students, Finch and Holmes, came with me to Cambridge when we moved here in what year did we move here? 1962, which was the year of the Nobel Prizes. And so we continued the work, and Holmes went off to Germany to be a professor of crystallography at Heidelberg. John Finch stayed with me, and Holmes gradually worked out the three-dimensional structure of the hemoglobin. But we had an outline of the structure as early as 1958, which is the model on the stairs.
So what were you doing? Did you come back to Trinity at that point?
I came back to Peterhouse as a teaching fellow at Peterhouse. John Kendrew was the director of studies. So at Peterhouse, afterwards I succeeded as director of studies, but I taught a great number of subjects because there weren't many teaching fellows. I taught crystallography for 1 year. I taught microwave spectroscopy and chemistry, but I always taught physics. And I taught physics right up to part 2. So it was rather odd. I was theoretically a Nobel Prize in chemistry. I went on in chemistry. I worked in a biological lab and I taught physics. I enjoyed the physics. I was quite a good teacher. And I think I may have told you this. Ken Holmes, when he came to it, found he had been taught by Fred Hoyle and what's the name of the very famous Indian physicist, Louise Johnson's husband, Abdus Salam, and he never learned anything. I said, how's that? He said, well, you see, they simply would dash off the problem. But you see, when I was a good teacher, because I had to work my way through it, learning it, I think I was a good teacher.
Did you enjoy teaching?
I enjoyed teaching physics, yes. I had a sort of strange double life. But it stood me in good stead because before I developed three-dimensional image reconstruction, I did various optical things in 2D using optical filtering, optical processing, which I wouldn't have done if I hadn't been teaching optics. So I enjoyed teaching. I enjoyed.
Did you lecture as well or not?
I gave occasionally I was asked by the university to give a course of lectures, and I gave a number, 6 lectures one time. Because Perutz was the one who gave most of the lectures, but one year he didn't give them, so I gave them. And then later, when I introduced three-dimensional electron microscopy, my method, I was asked to give a few lectures because it always seemed a bit bewildering. So I gave a few lectures in one of the biological departments, not the Cavendish, I've forgotten, it must have been physiology and electron microscopy. So, but I went on teaching at Peterhouse, right, and the Nobel Prize was in 1982. And I went on teaching to about 1984. And Hugh Dacre was the master, said to me, you know, if you want to, we can make you a supernumerary fellow, you don't have to teach. So I said, well, I'll become a supernumerary fellow, but I'll still teach for a bit. And I did for a year or two until I got too many other things. I'd started the zinc finger work by then, and so I got too involved with it. And in '86 I became head of the lab, so I was pretty busy. And president of the Royal Society in 1985.
For how long?
5 years.
What was that like?
Well, that was pretty interesting. I had turned it down 5 years before when Michael Atiyah did it. And it turned out I was the only person who refused it since Faraday. But the reason was that I just started a new division at the lab, and I thought being head of the lab was just as prestigious as being president of the Royal Society. Not in the wider world, but in the specialized world that I know. I think it did count for something. And I introduced the Department of Neuroscience here, which we hadn't had before. I did a number of things. So but president of the Royal Society was, I had to deal with a lot of issues. GMOs, genetically modified organisms, which, by the way, with zinc fingers you can do much better nowadays. In fact, it's producing what the agricultural scientists call a game change in plant agriculture. The zinc fingers are used to modify genes, put genes in. You can put genes in to a specified place, no longer just adding a gene and it comes to, wanders around and gets fixed in the genome in some place where it comes under inappropriate control. And so it's big stuff now. This is what's being done at Sangamo in plant agriculture as well as in therapeutics. So I wandered off. But then I spoke. So I had privatization from Mrs. Thatcher, which I had to oppose because she wanted to sell off all our laboratories. She believed that in the public sector you were lax, slow, all that sort of thing, or it wasn't like that at all. It was only in private industry could you submit to discipline, you see. So it was really an insult to this whole lab, and not only this, but others like it. And so I had to fight that. I also started on global warming. It was very obvious. And in fact, I did every year in my anniversary address, gave once a year for 5 years, I began to be like the elder Cato. On global warming, I said this, whatever the subject was. I think I said the trouble is I found most of the people didn't know who the elder Cato was, let alone the younger Cato, Cato Utica. So
you would thunder out dire warnings.
Yeah, well, I didn't thunder out; it's not my style. It wasn't said with huge. I did, but I did start the. One of the things about the human genome, I started the work here with John Sulston. Sidney Brenner and others were going around the world creating Hugo, just called the Human Genome Organization, and they kept going around having to know about better and better methods. But John Sulston, I inherited from Sidney, simply started out using whatever sequencing capabilities there were, and he made a huge start on it. And I encouraged him. Sidney wanted him to do the products of the genes, that's the proteins produced by the genes, and to sequence those, or to sequence the RNA, which is the intermediate between the DNA and the, so you could work with the, so you had sequences of all the proteins. I thought that wasn't the right way. Once you're sequencing, and with John Salson's techniques, well, he showed you could do about a million, a run of a million within a relatively short time, within a few months. So I encouraged him to do the whole genome, because there you get not just the products of the genes, but probably equally important, the regulatory machinery. You see, these are the upstream and downstream sequences, which are attractive factors which switch on the genes and make for specificity. So you get the control of the gene. You really understand the regulation, which is important as mechanism. A lot of the fellow scientists and scientists between a mechanism and a regulation of a mechanism. And so I think I once had to explain to somebody how a governor worked. You know how a governor works in a steam with the balls flying around. The regulation. So the word regulation wasn't in anybody's vocabulary. That's how you get, there's a word for it, shifts in
paradigms.
Paradigm shift, yeah. I wouldn't claim to be as great as that, but it was. I emphasize regulation. And now I'm working ironically on zinc fingers, which are the most powerful weapon for intervening in gene regulation. I didn't know that at the time. But anyway. So that was the Royal Society for 5 years. I had turned it down, as I said, but I was told, I didn't think they'd ask me again because I was already 69 years old. It was pointed out to me that Ali Todd became president at 69, so there were precedents for it. So I became president at 69. And my wife was very good because she enjoyed the challenge, and because we opened up the place, we had starting having lunches, and so Michael, Lily, Michael's wife, I don't know if you ever knew her, wasn't very, she played no part of the Royal Society, but my wife changed a number of things, first of all by making it slightly more social, and also by changing the food at gatherings. There was always canapé, she made sandwiches and things and proper food, and at meetings, so but it was, I thought that being in London, we had a flat in London, a very good address. I thought we'd go to the theatre and go to the galleries. I was so busy, there was things on almost every night. And so I did that for fun. And it overlapped with my being head of the lab, so I was pretty busy. So we had a driver who drove us into London and took us back again.
Coming back to the lab, you were working in the lab before you became head of the lab for quite a long time, presumably.
Oh yes, I came here in '62, and I was in the lab.
Who were you working with? You worked with Crick, or?
No, I worked, well, I had my own group, but I did spend some time working with Crick on chromatin. We published very few things together, but Francis began saying we should tackle our DNA, to work on chromatin, how it's packaged, and he had made some fancy models, but none of them were correct. So we had to get evidence for it, and I had a very good postdoc called Roger Kornberg, who got the Nobel Prize last year, who came with me, and we knew there has to be some substructure in chromatin. I knew this from the Xray diffraction we had of specimens of chromatin, and so he discovered, by doing a very good chemistry on the chemical analysis of chromatin samples, that the histones, which are used for packaging the DNA, on their own form aggregates, and the aggregates, there are 8 histones form an aggregate, and then it's at the end of my Nobel lecture I described this, so it's sort of tumbled it. There's a H3H4 form a tetramer, H2A2B. These are names of the histones form 2 dimers. And so the psychological breakthrough was this, that if the proteins form, a globular aggregate like hemoglobin, although a more complicated one, they can't possibly be sitting in the grooves of the DNA, which are people that thought Wilkins had spent years working on, you know. And then he cottoned onto it, and he made the basic step forward that the DNA must be wrapped around the DNA, and that's what he discovered the nucleosome. I didn't put my name on the paper, which I might have done, but because I got him to see that there had to be something repeating on a scale of 100 angstroms, and so he thought it was a linear chain, beads on a string. In fact, it's not beads on a string, it's a string of DNA on beads, beads of protein. I tried to push that forward. I couldn't make any progress with a string on beads. Nobody knew what it was. But still, anyway, so that was the chromatin work. But then I started some other things while I was here. I started tRNA. I also started an Alzheimer group, which is flourishing. And because I felt we ought to be doing something which is relevant to medical research and medicine. And I hadn't realized that Alzheimer's disease was specific to certain areas of the brain. I thought it was a general degeneration of the brain, but it's not. It starts off in certain parts of the brain called the entorhinal cortex, then it spreads. And so I realized it must be some specific malfunctioning of part of these. And so I decided we'd find out what it was. Now, it had been in the hands of neuro physiologists, and they'd been cutting sections of Alzheimer brains. I said we must get the material out, like we've done with chromatin. So I introduced chemical separation methods which were used on chromatin, chopping up the material, the enzymes, then fractionating them on columns or else on sucrose gradients. And so we discovered the filaments, which are abnormal filaments, which are composed of a normal brain protein called tau, which is abnormally assembled but chemically modified. So that was quite a work I feel quite proud of. It's been taken up and I have colleagues here working on it. But by then I'd moved on to zinc fingers later on.
Can we come back to, there were two things. One was, I don't know what his position, whether he was head of the lab, but you must have known Max Baruch's.
Well, Max was yes, of course I knew him very well. Yes, he was the head of the lab when I came here.
Yes.
And I was a group leader, that was my title. And John Kendrew was the head of of one of the divisions in the lab called protein crystallography. But Hugh Huxley, whom you may know of, he'd been a fellow of King's at one time, he was working on the structure of muscle. And muscle, of course, is complex. It's made up of many different proteins, actin, myosin molecules, which interdigitate. I was working on biorhythmic structures, are complex. They weren't just, Perutz and Kendrew were working on single proteins, myoglobin. So we worked on what we called biological assemblies, and we were using both X-rays and electron microscopy. And Hugh Huxley was the best electron microscopist of his time. And the way you looked at tissues that you cut, you froze them, you cut with a diamond knife or a very sharp glass knife, you cut sections, you built up a three-dimensional picture by a series of sections. Hugh Huxley should have had a Nobel Prize for the sliding filament theory of muscle. It's a great mystery why it never happened. After my Nobel Prize, I was allowed to nominate him year in, year out. It never came about. Speaking to one of the people I got to know on the Nobel committee, said it was a mistake, it should have happened, but it's too late now. Because there's no, to understand how muscle, a fundamental physiological thing, how does muscle contract? People had thought originally it'd be contractile proteins, which like a spring. There's not, as families of interdigitating filaments. It's totally unexpected. So there are some injustices in the world, and that's one of them.
What about Max as a character?
Well, Max was Max was a very single-minded and determined. He wasn't very learned, but as he went along, he learned all the different things that he had to do, and he wouldn't call him highly imaginative, but he was very determined, and he solved over a period of years the structures of hemoglobin, both in the oxygenated form, when it's fully oxygenated, and the deoxygenated form, and showed, as people had expected, a structural transition between them. The whole molecule changed its structure, and so it's able to, after hemoglobin carries 4 molecules of oxygen, and it's, as you go, 1, 2, 3 come in a certain order. When the 4th one goes on, the structure completely changes. It's a conformational 3-dimensional. It's a tetra, and it changes the conformation. It's a gross change, and Max gave it a very good title, molecular lung. It's what happens in the lungs, you see, molecular lung. So Max was, I wouldn't call him he wasn't one of these highly imaginative people, but he learned to do the right thing at the right time, and he stuck with it, and very lonely pursuit. It was 22 years of heartbreak, he once described it to me, when things didn't work out. John Kendrew, his colleague, very, very different.
What was he like?
John Kendrew was a marvellous staff officer. I mean, his own, very well organised. He kept records. Max had bits of paper. Absolutely.
You said he threw everything away, Max.
Max threw, yes. He didn't keep things. John Kendrew had a meticulous filing system. I'd never seen one before. He keeps all his records of music. It's a system of cards with holes in them, and you write on them. And then when you want something, you send. It's in a drawer.
Oh, yes.
And you send you have some long metal sticks which you put through, and you put them through the right ones. Then the card comes, all the other cards, your card can be lifted up.
I used to use those.
You used to have that. I know what it's called.
No. Something Cope Chat punch card system. Like
yes,
that.
it was before the computing of punch cards. But so Max was, but you see, Max was when I told Max how tobacco mosaic virus assembles, I don't know if it's in my Nobel. Have you got a copy of my Nobel? When I told Max how TMV assembles, he sort of nodded his head. Here's my Nobel lecture.
I've got it over.
You haven't got it on you?
Yep, I have.
Oh, virus. And this is, so I told you about nucleation and growth. So that's nucleation. No, it's the wrong model. It doesn't work like that. But you see, it enabled me to do experiments in which we started the assembly, making this protein disc, which you can make under certain conditions, and then you add the RNA, and the whole thing happens in 4 to 20 minutes rather than 24 hours. So because I separate nucleation and then the growth. Now I assumed that growth would occur by further discs going on and then dislocating and adding on. It turned out it doesn't go like that at all. It goes like that. Which is the most unlikely process. The disc acts. This is the RNA of the virus folds up into a secondary structure, and there's a loop there, and on the loop you have certain bases which get recognized on the inside, and it gets slurped up, and then you add another disc and there's more slurping. And what Max said to me, you're always telling us the most unlikely stories. Max, as I say, wasn't one of those highly imaginative. Francis saw at once that it had to be right. But the reason I'm telling you this is because before, when I was working on the structure of spherical viruses, which I don't mention here, I had said that the spherical viruses must be built out of hundreds of subunits, and they had to have a certain symmetry. but you can't build more than 60 units on the surface of the sphere. 60 identical units. You see in TMV, the key thing about TMV is all the units in the helix are identical to each other and also identically organized, identically situated with their neighbors. So that's called, that's because they have helical symmetry. So when we came to work on spherical viruses, which I gave, Roslin thought that I ought to have a problem of my own. So I began working on spherical viruses, and these are spherical shaped viruses with the RNA or DNA on the inside. And it soon became clear that many of them had more than several hundred units. And how do you explain that? Because you can't build a structure, you can't build anything more than 60 identical units on the surface of a sphere. Having 60 is the maximum number allowed by mathematical symmetry. And we had known that there had to be several hundred. And so I worked out, I developed together with Caspar, I introduced the term, the idea of quasi-equivalence. In other words, protein molecules in the aggregate don't have to know anything about mathematics, they have to reduce the energy. So if you can build a shell and you can deform the protein slightly, you will build, and you can build something with 160 units. And this was to prove to the doubting Thomases. This is the model of the virus which I showed you in the BBC. And it's built out of, I designed one just to show you could do it, out of little units like this. There's 180 units, and it's so arranged, if you see here that these bonds here aren't in a plane. So if you start putting them together, you get this. So it has built-in curvature. And with this, it's so arranged that the curvature will result in something with 180 units. So this is the complete one. That's a fact. But because you can't put on more than 60 identical units, you have to allow some play. So I put in some play, which in a protein will be taken up. So these things are slightly movable. And the bonds are slightly, you can change the bonds. So as you build it, you have to keep changing it. So this proves that you can do it. And but Max, so I didn't know that when I was doing all this work and put forward our theory of quasi-equivalence for virus shells, for spherical viruses, no, it's not equivalent, but quasi-equivalent. Max sort of shook, and I used to explain to Max, used to say, yes, very interesting and so on. And I didn't realize until a few years later he didn't believe it. And I'll tell you how I know this. He, in 1966, he gave an interview in Science magazine to a reporter. And Max spoke about all the successes of the lab, the Nobel Prize in '62 for Crick and Watson, and himself and Kendrew. And Brenner's work was starting to come out on the messenger RNA. He also says in Klug's work, it's very satisfying. He said it was very far-fetched, you know. Now, he never, when he was director, he never let on. I used to tell him the stories, he would shake his head as though he was understanding. I didn't know he didn't believe a word of it. But you see, it was his gift as being a director. He let me get on with it. He didn't try to tell me, persuade me this was nonsense. And Crick understood it immediately. In fact, I know that Crick put me up for a Nobel Prize. He mentioned the spherical viruses. It wasn't mentioned in the theory of, they like Buckminster-Fuller geodesic domes. See, but Buckminster-Fuller, and you have to have 5s and 6s. If you notice, there are rings of 5, rings of 6. And you have to have 12 rings of 5. It's like a football. The people hadn't seen footballs in those days with hexagons. You know, football. The Greeks made balls out of goat skins, and they sewed together 12 pentagons and 20 hexagons, and that made football. Which was, and then they started making football. When I was a boy, the footballs were made of strips. And then the modern footballs are hexagons and pentagons, but they hadn't existed then. And so Peirce was a very good director because he let people get on with things. and he didn't try to divert me from what he thought was a blind alley.
Can you very quickly, this all overlaps with the work you got the Nobel Prize for, but if you could explain very simply the technique, which you said wasn't crystallography, but it was three-dimensional.
Well, electron microscopy.
Yeah.
Electron microscope is a very powerful way for imaging, and it was invented in the 30s. But in the electron microscope, unlike the light microscope, it can't be focused, so you can't focus down on a level. So when you take an electron micrograph, everything in the line of view is projected into a 2D image. You see that?
Yeah.
So how do you unscramble it? Well, I unscrambled it by, very simply in the end, by taking the specimen and rotating the specimen so you get a series of views from different directions, or else by looking at a random field of particles. I can show you this. A field of particles in which they're lying higgledy-piggledy in all orientations, and are trying to relate the different views to each other. And if you can, if you tilt systematically, you know what the angle is between. You know what the angle is. I'm just looking for. I need a chair.
All right.
Image reconstruction, we call it, because you combine all the views mathematically, but we're using a computer. I have to explain that to produce a three-dimensional image, and it's the basis of the X-ray CAT scanner. Some people think I should have got a Nobel Prize for X-ray CAT scanner because they copied it. But they didn't. The patent, Hansfield, who got it, when he took out the patent, didn't mention my work at all, but he patented the machine, not the technique. So I felt a bit sore about that in 1979 when they got the Nobel Prize for the X-ray CAT scanner, because he knew my papers, referred to them, and I'd exhibited with him at the Royal Society. Anyway, I'll show you what we used to do. Well, so we had a theory of how you might build these virus shells or the quasi-equivalents, and so this is the structure of these are warts. These are warts. Human wart is papilloma. Papilloma is rabbit papilloma. Polyoma is another wart-like tumor disease. And if you take electron micrographs, and here you see, I figured out from just by examining the X-ray, the EM pictures, which look like this. This is a field of pictures. Here's a field. These are virus particles, and this is the surface. Here they are lying in all orientations. So what I did was to introduce tilting experiments. So if you look at particle, here, this one. Where's the threefold? Oh yes, look at this one. So you see here, for example, are different views. This is particle A, and these are models. We're modeling it on a computer. And here we have a whole gallery of views. You see, you're looking right through the object, top and bottom. That's very confusing. But in the old days, I figured it out and showed how. And so we made, this was all the steam age before I introduced three-dimensional image reconstruction. And here you see how we build it. We build the images. Let me just get this. Yes, here we have model building, you see. So here we have one side of the model. Here I'm putting in, do you see the hexagons there? And there's some pentagons. This one has 60 hexagons and 12 pentagons. You always have to have 12 pentagons. And so these are, now this is changing the size of the hexagons and the pentagons. One side of a molecule, you see here they're getting bigger, here they're fusing. And then if you see the top and bottom together, the two sides, and in certain view it looks like that. You see, and so we look at the electron micrograph, and here are the views. Here are selected views, and we can see this group of three, and there it is simulated. Here. And so all this was sort of steam age stuff, and so this is how we solved the structure of a number of patterns. And later I introduced, and I realized that here we were deducing the structures from a series of different views. So there must be some way, the information is there. Oh, sorry, the information is there. Construction. So here you see a model, it's a 2D example going to 1D. And if you have an object like this, and those are holes in the structure, and you take views in different directions, what you'll see are these. Now if you use Fourier analysis and you make Fourier transforms of these in 2D, these three can all be combined to produce the original object.
Amazing.
But it was, yes, and so this is the basis of the Xray CAT scanner. And so that was the, so you see, and the method of Fourier analysis, you can actually work out, depending upon how much detail you have, the number of views can be determined. So the finer the detail that you want to get in the final reconstruction, you have to take more and more views. And there's a simple mathematical formula which I worked out. And this is what it was. So as you can imagine, when Hounsfield my paper appeared, I think I may have said this already, appeared in January 1982. I mean, sorry, what am I talking about? January, I'm getting all wrong the dates. In January 1968, in August 68, I may have said this already, he took out a patent for building a machine. He got EMI of having 60 detectors, and you put in the object as the patient or the X-ray, and you took these views, and then you combined them mathematically, using not Fourier transform, but some rather crude version of it, which the first time it produced nonsense, because he didn't have enough views for the sort of detail he was looking, and but then in that was 79, 82, I got the Nobel Prize on my own for chemistry. So that's but Max. So that's the basis of it all. So X-ray would be called computer automated tomography, and I had quite an interesting time trying to get the radiologist to take it up. I told you the story already. So when I realized this, I went to see a professor of radiology whom I knew. Well, I knew his right-hand man, and said, look, instead of taking X-ray pictures, you should be able to say, he says, but if you take views in different directions, you'll give the patient a lot of radiation. I said, but look at the detail you'll get out. I didn't at that stage know that you could also see different levels, so you can distinguish the density of a tumor tissue from an ordinary brain tissue. That I wasn't able to show that. But I knew you could make a three-dimensional picture with different degrees of gray levels. That's different degrees of, you know what I mean by gray levels. And so, and one of my colleagues went to the radiologist here, and they said, look, we don't need all this fancy stuff. We know exactly what we're looking at. And I said, but look, how do you find the detail? He said, well, we do computer tomography. What's that? Instead of taking a simple X-ray picture, you have, and if you ever had an X-ray where you lie down, and there's the X-ray source moves along at a certain rate, and underneath your back there's a camera, as a film, and the film moves at a certain rate. So depending upon the relative motion of the X-ray source and the photographic plate you image in inverted commas isn't focused, you get a certain level, at a certain level in the line of view. Everything's in geometrical alignment and everything else is blurred, and they call that X-ray tomography. And that's what they do. But you see, later I was able to show them that you have an enormous amount of wasted, because you have everything above and below the plane of view is blurred. In fact, you're using much more radiation than you did for the for the information you get from a three-dimensional image reconstruction. So I introduced that. So I felt a bit peeved when I didn't share the Nobel Prize with Hounsfield and the others. But I was busy with other things, and I think as in '82. But you see, I think I may have said this. The citation for the Nobel Prize called it crystallographic electron microscopy, which isn't at all enough crystals to do with this. But they use this Fourier analysis, and they didn't understand what Fourier analysis was. So I can do mathematics, but I call this applicable mathematics. The pure mathematics would laugh at what I do. Applicable mathematics.
You then went on. I mean, you've had a whole new set of work. Could you explain this work, which you've alluded to a lot of time, which is zinc finger work?
Well, yes. Well, it's in my introduction. Did I give you this? Yes. This thing tells the story. This is I was asked to write a well, it says how I came to do it. I became interested in active chromatin. That's chromatin, which has already been, the DNA has already worked itself loose, or it's being worked loose in some way from the chromatin. It's about to be transcribed. That is, the regulatory proteins which are going to switch on the gene can get access to it. And that you can show, this active chromatin, because it's now become susceptible to enzymes, DNA enzymes, which will attack the DNA. So you found, if you looked at chromatin, which you extracted from living cells, you'd find that some of the chromatin was accessible to enzymes, and those correlated with, once you knew what they were, these would be genes which were going to be activated. So I began looking for a source of active chromatin, and now and I wanted to investigate that. It's described in the introduction here. So I read a lot of the literature. Now, if I was going to do structural work, that's X-ray crystallography, biochemistry, and possibly electron microscopy or things of that sort. If you're going to do that, you need large quantities. So I looked for something which you get milligram quantities out. So you can do a lot of biochemistry using microgram quantities. This is because the proteins are run on polyacrylamide gels, where you only have very trace of proteins, and these show up in what's called gels at apheresis. So I wanted something that you could get quantities on to crystallize or to investigate by serious biochemistry. And so I found that there was reading literature, there was a gene of in Xenopus, the South African frog or toad, which was present in large amounts. I was alerted to it by reading various papers, and one of my colleagues, Hugh Pelham, had actually worked on it, my current colleagues, and he showed me some, and I see if we get large quantities from it, so I decided to work on the 5S RNA genes of Xenopus. These make 5S RNA, which is the product of the gene. In this case, the 5S RNA gets incorporated into ribosomes, which are protein synthesis factories. That's by the way. And so I decided to try to get to work on it. And I had a new postdoc, a young man called Miller. I put him to work on it, and I discovered, or uncovered, by a series of purely biochemical experiments over a period of several years, that this had a repeating structure. There was a repeating structure in the and this was the repeating structure, and it had 9 units, and this is the DNA, and these 9 units bound to the DNA. And so what we knew at the time is that they had certain cysteines and histidine residues, C's and H's here. So I interpreted it as folding up into little units, and each unit bound a certain number of bases. And when the sequence came out, the protein sequence, the amino acid sequence came out of the zinc fingers, here you are, there are 9 of these repeating units, and these are the 1-letter code for amino acids. And you see they have 2 cysteines and 2 histidines in each of the units. And these are the ligands. These are the commonest amino acid which bind zinc. And we measure the amount of zinc, and there was between 7 and 11, and there are 9 of these. So it was all done biochemically. Later on, people thought it was all done by bioinformatics. It's a load of coswallop. It was all done by chemistry. But people couldn't grasp it. Still people are writing it was all done by bioinformatics. So it went on from strength to strength, and this is the most common, it's the most common motif. And here you are, the total number of genes. 3% of the human genome consists of cysteine 2, histidine 2 times zinc fingers. There's another type of zinc finger called C4, which I won't go into now. And you see, as you go up in the evolutionary scale, you get more and more of these. So down in the C. elegans and the worm, and the zebrafish, you get 1.7%. In the human, you get 3%. So you see, they're a marvelous modular system. It's a combinatorial system. You can combine the fingers. Each finger has a different amino acid sequence which can recognize the sequence of DNA. And so this became a tool for, so it became the ideal tool for making artificial synthetic fingers for having access to genes. This has become a big technology. Did I send you any of the press releases?
You sent me one, yes.
Yeah, well, that was the, so now it's in patients. And so it's a really major, well, it's a major piece of work, and it's taken some years to, I started out doing it myself, but we formed well, my colleague Yan Chu, who was half Chinese and half Greek, very entrepreneurial. He left the lab, he could have stayed on here, to form a company called GenDAC. Have I mentioned this before? GenDAC, Gen for gene, DAC for dactyl, Greek finger. And he founded the company and got some backing from it. And because the MRC, we took out patents, I should say, because we took out patents, and the MRC held the patents. And in the lab, we started making libraries of zinc fingers. We began to work out the rules of recognition. And this GenDAC was bought out by an astute American who created a business. Yan Chu created a research establishment, very high class, very high powered, but no money, because people didn't invest in it. There were a few backers from an MRC seed fund, and so Sangamo, which means Sango doesn't mean anything. It's named after Sangamon County in Illinois. His grandfather had a business there, and so I go to the States 2 or 3 times a year, and they now going great, as they would say. They might even make some money. It's now it's become the method of choice of. It's also now in plant agriculture, and it's going to make the old-fashioned GMOs look like steam engines compared with the internal combustion engine. And it's being developed by Dow Agricultural Sciences, and the American company, and they say this is game-changing technology. That's high praise indeed.
Paradigm shift, indeed.
Paradigm shift, yeah, game-changing. Yeah,
excellent. Well, congratulations on that. And
well, I haven't. Yes. So
well,
I know I've been nominated for another Nobel Prize for it.
I've heard that too. So
it may be. Well, it depends.
It'd be wonderful if I had the 2 living Nobel Prize winners in the world. I've interviewed Fred Sanger, and if you get a second one...
Well, I wouldn't bet on it. I've got to live long enough. They're hard-headed, you see. And how do you know that if you make a cure, it won't revert? So I hope I live long enough to see it.
It sounds as if you will.
Yes, so that's how I came to do it. But I was purely following. I didn't start out, I think as I've said so in my Nobel addresses, I didn't take up science in order to be a benefactor of mankind. It's curiosity. And that's what the driving force is, curiosity. And it's not only thinking, you see, I know that you thinkers, but it's also doing. And I put a lot of work into doing it, you see. You can't...
When you say doing, do you mean making things or...?
Yes, we used to make them in the lab. We used to make fingers in the lab, chemically. And then so I had to learn how to synthesize these things. And I know enough chemistry. Well, you know, my PhD is in physics.
Yes.
But over the years I've learned enough chemistry, and I don't do it with my own hands, but I know what to do. I understand the chemistry. And so that's what I mean by doing. And it was only because of that Sangamo... Well, what happened in 1994, I discovered the zinc fingers in the first paper was 1980. I think was the first paper. 1985. That was then. It said it was a. I should give you an original, but I haven't got any left. It was a DNA binding repeating unit in the gene of something or other. You see, of a xenopus gene, and so repeating, and so the. And that showed, and it does say in the end that this is a very beautiful combinatorial scheme. It's unlikely to be confined just to this, I didn't write this, but I used to say, to a lowly gene in a lowly animal. But you see, the whole thing is that this happens to be what we're working on. People could have discovered it in somewhere else. And so well, you know what Pasteur said, chance favors... I used to think chance favors the prepared mind. No, but somebody told me it's the wrong quotation. Chance favors only the prepared mind. And I haven't checked that that's the true, but it's more true that it's only. You know, because you have to be thinking about something in order to. But what are you prepared for? You see.
Yes.
So
and you said it wasn't. When we were walking down the stairs, you mentioned it wasn't just intelligence, very cleverness, but some imaginative...
Yes, some imaginative powers, yes. And I think I've got a... I think I do have an imagination. There's some people say three-dimensional imagination. No, it's imagination is what if? What if? What if? It's that kind of imagination. And you also have to have some technological capacity in order to do it. You see, otherwise you can't sort of say, you can't just write like the great man Pauling when he was going to solve crystal structures. You know, the great Linus Pauling.
Yeah.
He would write at Caltech when they tried to solve proteins. He would write, his eye occurs a lot, I have decided this is the way to do it. He would send this out to the man at Caltech. No, and he never said, he proposed things which were unrealizable, you see. He didn't understand enough of the detail. The truth's in the detail. That's a quotation from Lucien van Leeuwen. You ever read it by Stendhal? It's quite one of his lesser known works. There's a marvelous sentence which I've got in French somewhere, but what it says is, tell me, he said to his uncle, tell me it all, but tell me it's in detail. There's no truth but in anything but the details. I'll find the French quotation. I looked it up in Stendhal later. I read it in English. Lucien van Leeuwen. One of those.